going to start going over human immune system and how your body reacts to an infectious pathogen when it gets into your system where it's not supposed to be. Last time we talked about all the possible kind of characters that are involved, antigens, markers, the pathogen-associated molecular patterns. Talked about blood, different blood cells, white blood cells that are involved, lymphatic system, the function of it. So now we're going to look at the human defense system against pathogens. Typically, most books organize it in this way. You have the first line of defense, second line of defense, and third line of defense. Each of them have a purpose. Each of them are either specific or non-specific. We're going to use the same way to go through it. So first line of defense. This is basically it's an external defense. The purpose of first line of defense is to first not even allow a pathogen to get in. The best way to fight is to have an offensive move which prevents the enemy from getting in. Okay. So first line of defense is non-specific, it is innate, every human is born with it, and it is external. Meaning, these are found in the external areas of your body where it's exposed to the external environment. They're mostly your barriers. So their job is to prevent the pathogen from getting in to begin with. So if you don't have an infection, your immune system doesn't even have to get started. Skin, mucous membrane, and some other protective barriers that we have are the ones that are found in your first line of defense. Skin. It is one of the largest covering tissues in human beings. Your entire body is covered by it and separates the external environment from your internal environment. So lungs, heart, all of your abdominal cavity, thoracic cavity, these are separated. For it, we have the epidermis and the dermis layer. Okay, so we're going to look at those. Each of those those are the main parts. Epidermis is this little top portion, and then the dermis layer is right here. We're going to look at how they're designed in humans to provide protection. First, we're going to look at epidermis. This is an electron micrograph of a human skin, just not even enlarged. I have an image of an even more enlarged one. This is what the surface of your skin looks like. Epidermis, first off, it is composed of a multiple, thick, packed, multiple layer. The layers are overlapping. So this is an enormous, it's an extremely enlarged human skin. This is how you can see why we're related to lizards. Okay? So overlapping, tightly packed overlapping layers, meaning the layer underneath, this is the gap between the cells. The next layer is going to cover the gaps. And they're very tightly packed. In addition to that, the top layer of epidermis is composed of dead cells that are constantly sloughing off. We're constantly shedding them. The live layer of epidermis is actually at the border of epidermis and dermis, and it divides and adds cells to the top. The top layer is composed of the dead ones. How that protects us is if any pathogen is on the surface of your skin, by sloughing it off, you're going to get rid of it. You're removing them from your skin. In addition to that, in the epidermis layer and the dermis layer, we have the fixed macrophages in this area. Remember, fixed macrophages that are found in the tissues, we refer to them as dendritic cells. So the ones that are found in the skin, either they refer to them as dendritic cell or Langerhans cells. Both of them are fine, but they are fixed macrophages. And their job is to constantly intercept invaders and take a look at them. So these are your fixed macrophages. Remember the old image that I showed you, the macrophages again, when I had the fixed macrophages. So they're constantly roaming in the middle of the tissues at the surface in order to check for it. Now, the next layer is the dermal layer, the dermis layer of your skin. This is where you have your capillaries, this is where you have your sweat glands, your sebaceous gland or your oil glands, where the neurons are, all of that are found in the area. Dermis obviously provides a lot of nourishment for the epidermal layer. So that's one way, by protecting your epidermal layer. The other thing that is made in the dermis layer is, this is where you produce most of your collagen. I didn't put it up there, keratin is another protein that is very similar to collagen, hyaluronic acid. So we're mostly focusing on collagen here. What collagen does, it's elastic, it's protein made up of protein, and it makes your skin very elastic. So the more collagen you have, the more bouncy your skin is, and it's much more difficult to break skin. When babies are born, they have so much collagen. It's not because of how much fat they have. If you try to lift their skin, it's almost impossible. It bounces back immediately. As we get older, we can do this a little bit more, and it takes a little bit longer to go down. It's cruel, but if you have somebody who's around 80s, when you do this to their skin, it stays up. It takes forever for it to go back. Like when you do this, it stays like that and very slowly goes down. That's because they lose so much of their collagen. So it doesn't bounce back. Because of that, that's what they call, like, their skin gets thin. It breaks easily. When you do that, you can break it because it can't bounce back. Sweat glands provide us with some protection too. Sweat produces salt. Salt creates, and is released to the surface of our epidermis. It creates a hypertonic environment. Not every microorganism is going to like that. That provides an hostile osmotic environment for them. In addition to that, we produce an enzyme called lysozyme. So do not mistake this with lysosome, S-O-M-E, which is our organelle. Lysozyme is an enzyme that breaks down carbohydrates. A lot of bacteria have carbohydrates on the surface. It can start breaking it down. Fungi, cell walls that they have, carbohydrates, it can break it down. So we have that in our sugars, in our sweat. We release it. Then our sebaceous gland, oil glands, they produce the oil or sebum. Oil does two things. First off, it moisturizes your skin. It makes your skin much more moisturized. If the skin is moisturized, it is less, when it's dry, it breaks easily. But when it has the moisture, it doesn't break. So it reduces the breakage of the skin, damage to the barrier. Second, the oils have fatty acids in there. Therefore, the pH of the surface of the skin decreases slightly. So it is in the pH, lower pH ranges, so it's acidic, which makes it, again, a little bit hostile for most microorganisms. Okay? So that was your skin. Not every area we have a skin. So you have internal, external environments. Your oral cavity, your upper respiratory, opening to the reproductive tract, opening to the urinary tract. The entire GI system is an external environment for us. However, you can have the skin because those epithelial cells and the cells that are connective tissue that underneath typically have a function. Like your GI tract, you need those skins in order to be able to release the digestive enzymes, release the stomach acid, absorb the nutrients in your small intestine, in your colon. So we don't have skin in that area. We have the mucous membrane or the epithelial layer. Epithelium is the part that is like our barrier in those regions. First off, the epithelium of mucous membrane is like your epidermis. It is made up of few layers. However, all of them are alike. But the top layers are loosely bound and they can be shed off. This is why when a person, for example, you've seen in these movies, the person takes a sip of a drink and they grab it fast and they're like, we have your DNA now. Chewing gum, anything, spit, they have their DNA. You just have to take a swab and rub it to the inside of your cheek because those cells fall, and in that person it's in their saliva. The surface of their lip, that top layer of skin, stays next to the glass. So all of that, they can actually get cells from it. But in addition to that being loosely packed, the other part is the mucus. So part of the cells in the area are capable of producing a thick mucus that covers the epithelium of these areas. The mucus is typically produced by goblet cells. That mucus again gives an additional protective layers. In some areas, like your lower respiratory, the epithelium is actually covered by ciliated cells. So this is the epithelial layer of the inside of your lungs. It is ciliated. Those cilia actually are constantly moving. See how they're all in one direction? So they are constantly moving in simultaneously in one direction. In your lungs, it's upper movement. We call that upper excretory movement. What it does, so this is the goblet cell, the ciliated cells. It creates a thicker mucus, and that mucus will trap anything that you breathe in that is not supposed to be there, and the ciliated cells basically move it up, and you will swallow it. Where your trachea and your esophagus meet, it comes off of that and you swallow it down your esophagus. It goes and you digest it. All right. In addition, in these areas, you have the Langerhans cells or dendritic cells. Fixed macrophages are found in these areas. Fixed macrophages also, for example, in your lungs, at the air sacs, you have the alveolar ones. Okay, so in that same area, those are the alveolar. So you do have them. The mucus and any nasal secretions or any secretions that you have will also have lysozyme, that same enzyme that will digest some carbohydrates. So that was your mucous membrane. There are some parts of your body that you have neither of them. The surface of your eyes, it still needs to be protected. Granted, you have your eyelids and then eyelashes. So you're constantly blinking. If you want, you can close it, protects the surface of it. But at the same time, when it's open, it's not protected. For that, you can have mucous membrane because it obstructs your view. So we have tears. We produce through lacrimal glands, we're constantly producing tear. That tear is constantly washing the surface of your eyes, and blinking helps it too. The tear then is drained down to from the back of your nose to the back of your throat. That's why you can actually sometimes, if somebody puts droppers in your eyes, after a while you suddenly taste a bitter taste of medicine, because the neurons that taste bitter is at the back of your tongue and you can taste it. Or some people with their interesting act, they can actually do the reverse and drink something that has color and bring it through their eyes. They've learned to do the reverse. So it cleans it and it goes to the back of your throat. Again, all of the protective measures that you have in your upper respiratory and the back of your nose and throat will protect you from that. Saliva. In your mouth, in addition, you have saliva. Your salivary glands are constantly producing saliva. And that's supposed to basically rinse the surface of your oral cavity. That's why at nights you don't produce a lot more saliva as you do during the day. Why? Because you will choke on it. So you reduce it. That's why you have more of plaque formation on your teeth at night rather than throughout the day. Your saliva has lysozyme. Your saliva has the enzyme amylase that breaks down starch. So all of those, again, help with the microorganisms. Some other areas of your body, stomach acid, it's highly acidic. It's hydrochloric acid. It's one of the strongest acids, so you're producing. It makes it semi-hostile for a lot of microorganisms. Then in many of these areas, you also have your normal flora. So your GI tract, in your colon, in your small intestine, in your oral cavity. We have a lot of normal flora, which, as we talked about it in chapter 11, they can protect you by antagonistic behavior against any microorganisms, by giving your immune system, forcing your immune system to be present in those areas and constantly checking, helping you with vitamins, all of that. So that's how it goes. So that's your first line of defense. First, let's not get anything inside. Let's even fight it before it gets inside. Obviously, as you know, it doesn't always work. There are some true pathogens that can basically bypass some of your normal barriers. And they can basically, they're not stopped and get through it. Or your normal barriers get compromised. You have a cut in your skin. Mucosal membrane, you're not producing enough mucus, something has happened. Again, bacteria, fungus, they can get passed by it. In that case, when your first line of defense does not function, we will have the second line of defense. The second line of defense is internal. Pathogen has bypassed your external barriers, now it's inside. Okay? Or it's even, in some cases like the GI tract, it's colonizing your colon. So again, disrupts the normal function of the body. Second line of defense, this is when your immune system gets triggered, gets an alarm, and starts reacting. Either because cells are damaged in most cases, or disrupt the normal function, so your immune system gets triggered. When your immune system gets triggered, it actually produces the second line of defense, which is non-specific, and we call it inflammatory response. Your inflammation. Whether the signal was because your barrier, you just cut your skin, tiny little cut, there is no pathogen even there, but your barrier is damaged. It has to fix that. It has to prevent any pathogen. That will cause inflammation. It's inside in your GI tract. That will cause inflammation of your GI tract. Inflammation in general, when you think of inflammation, this is what you should imagine. On your hand, inside your gut, inside your colon. So, redness, swelling, pain, and loss of normal function. Those are typical signs of inflammation. So in this case, this hand is hot, red, swollen, and it won't allow them to use their hand is not going to function properly. It's so swollen, they probably won't even be able to grab something, turn something, write with it. It's painful. So let's take a look at the process of inflammation and see what inflammation is. I'm going to use the animation in a bit to go over this, but few things we're talking about. We're just going to talk about inflammation. Imagine you have a cut to the skin, and whether pathogens have gotten in or not, but one of your barriers is damaged. So like they have it. What ends up happening is, let's say these are the cells of the barrier, all right? These are your cells, normal cells. Whenever you have inflammation, blood is associated with inflammation, and that inflammation is typically associated with the capillaries. So capillaries. 2 L's or 1 L? 2 is 2, yeah. Capillaries. So remember, you have, well, I'll remember if you haven't had anatomy physiology. Human blood vessel, we have thick arteries. They have multiple layers. They have thick muscular layers on it, and the blood goes through it. This is so when the heart pumps it, your arteries are supposed to basically grab that pressure, keep it, and pass it on and force the blood into all the areas. Arteries break down to little narrower ones, arterioles. Again, still they have like muscle tissue, connective tissue, multiple layers. But when it gets to the capillaries, capillaries have capillaries are made of single layer cells. So capillary is just one layer of epithelial cells. Why? Because this is where exchange is happening. You can't have muscle and all of that there. So plasma seeps out, oxygen, the red blood cells are here, oxygen goes to the tissues, carbon dioxide comes here. Any other red blood cells that you have in capillaries, they actually will go one in a row. They're so narrow. Your blood pressure actually is lower because it branches out, and so the process is happening. I'm going to use the animation. So what we are saying is, in the process, something has happened. So you've cut yourself, so some of these cells have been damaged. When the cell is damaged, cytoplasmic material are released. Cytoplasmic material should not be found in your tissues. DNA, ribosomes, these things should not be found in the tissue. So when they are, that tells your immune system, uh-oh, something's wrong. On its own will induces inflammation. The term induce or induction means it triggers it. It activates. Like, you know, when you say, a woman says, I'm going to be induced today, they're going to force her to have the baby. So induction, induce means you start a process. Cytoplasmic materials themselves start the process of inflammation. In addition to that, in most of these tissues, barriers, mucous membranes, you have mast cells. So, mast cells. Mast cells themselves, if they feel cytoplasmic material, they release inflammatory inducing chemicals. So, for example, histamine is the one that I said we're focused most on. So histamine, you guys have all heard of histamine. Oh, I have an allergic response, I'm going to take an antihistamine. All of these, most of these anti-allergy medications that you have out there, they have antihistamines and then maybe some other inflammatory inducing. So histamine is an inflammatory inducing chemical. Ultimately, all of these inflammatory inducing chemicals, they will cause the activation of inflammation. Activation accumulation of inflammation will cause a change in the capillaries, which forces blood to come to this area, a lot of plasma leak out, and white blood cells from leaking out of capillaries. So remember I said inflammation, think about swollen, ton of plasma into the area, all of these cells leaking out into the tissue. That thing is going to swell up because now you have more fluid, you have more cells that weren't supposed to be there. It's going to swell up. When you have more blood coming, you have more red blood cells. So swollen, redness, red, two D's? Two S's, okay. So one D, right?
Yeah.
This is why I don't care for, I don't take spelling errors out. Unless it sounds like it, then it's good. So swelling, redness, a lot of red blood cell is going to get there. So this is because plasma leaks out. This is because white blood cells are going to go out. Redness because you have a lot of red blood cells coming here. Pain. Pain, your body's reaction to tell you something is wrong. So, if you don't have a pain anywhere, if you don't have loss of function, you don't pay attention to it. Ultimately, loss of function. So, plasma. Why is plasma leaking out? Plasma is going to have some defense chemicals that it's going to help. Plasma gives you nutrients. When you have a damage to the area, you have to fix it, right? And when you fix it, you need more ATP. When you need more ATP, you need more nutrients to break down and get ATP. White blood cells, they have defense. They bring on defense. Red blood cells help with increase in gas exchange. When you're making more ATP, you need more oxygen, you're going to make more carbon dioxide. So pain is giving you alarm. Loss of function until everything is repaired. All right, so what we're going to look at is, first of all, what is happening here in the animation. White blood cell moving out is going to be as a result of all of these things are happening because of chemical stimulants, right? The chemical release causes inflammation reacting. Chemotaxis is how white blood cells know how to get to the area. They see histamine, they're like, I need to be there. So by chemotaxis, they're attracted to all these inflammatory inducing chemicals. So that's how they know I gotta go there. You will see inflammation will actually create exit signs for them, so they need to get out of here. We refer to this, and you'll see it, it's called diapedesis, movement of white blood cells out. So let me use the animation now to show you this process. I'm going to play the animation without the sound at first to show you the process. And then I will go over it. So it starts weird, but just the guideline. So this is the connective tissue. This is your barriers. Does the mouse show up good?
Yeah.
Okay. So this is your barriers. If you want, I can turn off the light a little bit. So better, huh? Okay. So this is the skin. They're using an example of you basically have a splendid there. You cut yourself. These little things are, the green ones are representing bacteria. Even if bacteria wasn't there, the damage to your cell will cause inflammation. But they're saying, let's say we even introduced bacteria to the area. These little star things are going to be your inflammatory inducing chemicals. So they are releasing there. This is the capillary. So see, single epithelial layer cells. This is inside the capillary, red blood cells, white blood cells, blood, everything is going through here. Got it? All right. Tell me to slow down anytime you want. Based on your class, on my Thursday class, I was going so fast, nobody stopped. I finished that lecture for you guys that I went in an hour and 20 minutes. I finished it for them in an hour and 5 minutes. So stop me. All right. So damage happens. Damage to this causes the cytoplasmic release of cytoplasmic material. That basically innate defense got compromised. That causes basically inflammatory inducing chemicals being released. Those are your cytoplasmic material. What they're not showing you now, and they will show it later, they're going to show a mast cell here. So these guys coming in the area will tickle that mast cell, and mast cell inside of it has histamines, prostaglandins, and all of interleukins, all of these different inflammatory inducing chemicals. So mast cell will add to that too. All of these are happening simultaneously. This animation is going step by step. Okay? Now, first off, what they're going to show here in this animation is the first change to the lining of the capillaries. So to this lining of capillary, what they're starting to show is the appearance of these little proteins. These proteins are called selectins. If you don't know it, for me, it's fine. Basically what it is the lining of your epithelium. Now these proteins that are there, and they're called selectins, they are going to get activated, get turned on. Kind of like in case of emergency, they say the lights for the exit is going to light up at the plane, and you just follow those exit lights. Selectins basically are communicating to the blood cells. This is where inflammation is happening. Because inflammation, if I've cut myself here, you don't want inflammation here. I want inflammation here. So all of your cells know this capillary is going to go through a change. The selectins are going to become active. This thing is like the alarm, the flashing hazard light, and says, here, this is where you want to go. All right? Selectins get turned on. Your white blood cells, on their surface, they have a corresponding protein that is called integral proteins. Again, you don't need to know. They're just showing neutrophils. But all of your white blood cells and a lot of neutrophils have on their surface these specialized proteins that are complementary or corresponding to selectin. When selectin becomes active, these integrins can actually attach to it. When they notice selectin, it slows their movement down and it attracts them. So they will attach to the surface in those areas. So these receptors attach and they stay there. This is actually called margination. So now all of your white blood cells, they're showing neutrophils here, but majority of your white blood cells have that. And they will attach to that little area of the capillary where the signal is going. This is where the damage is.
Now those are white blood cells?
Those are white blood cells. Red blood cells will not go out unless there's a major cut, but red blood cells tend to stay. They don't have a job inside in the tissue. White blood cells do. So for example, what they have here is neutrophil. So neutrophil can't do what you want it to do in the blood vessel. it needs to go to the tissue, do two things. Phagocytosis of the damaged tissues. So when the wall is broken and you want to build a new wall, first you have to get rid of all the broken wall material. So that's one of the reasons you have phagocytosis. The other one is, if you have invaders, you start removing them. So if you remove them, hopefully you won't have an infection, right? So neutrophils get there. At the same time that this is happening, other inflammatory chemicals, and histamine is one of the main ones, is also being released, and it will affect the epithelial cells, the lining of the capillary cells, a little bit differently. This time, as you will see, histamine, what it does, it causes your capillary to expand. So the diameter increases. The diameter increases, but the number of the cells remains the same. In order to increase in diameter, these cells will separate from one another. So the histamine is going to increase the gap between these single layer of the cells in the capillary in that area. That opens a gap that allows cells to go out. They're going to show the mast cell now. Margination. They're going to show histamine. So this is histamine. The purple is histamine. That's going to cause what we call vasodilation. So it dilates the capillaries. It expands them. The vasodilation causes an increase in the gap between these epithelial cells of the capillary. What that does, then after that, these white blood cells are like, fantastic. Now we have an area that we can squeeze through. They're going to squeeze, change shape, and go what we call diapedesis. They're going to crawl out. By the process of chemotaxis, they will go to where first the signals started appearing. They will crawl to the area. That's called diapedesis. So they're going to show neutrophils. Plasma is going to leak out. And then the neutrophils here in this image, they're going to squeeze, change shape, squeeze through, and get to the area. Then they're going to start getting rid of the bacteria, damaged tissues, and hopefully with a lot of growth factors, other cells that are involved, you can repair and maybe stop the infection. All right, let me now play this thing for you guys from the beginning.
The inflammatory response is an important innate defense against tissue damage. It begins when injured tissue cells release chemical signals that activate the endothelial cells of nearby capillaries. These activated cells begin to display adhesion molecules called selectins on their surface. These adhesion molecules attract neutrophils in the bloodstream, slow them down, and cause the neutrophils to roll along the endothelium. Pro-inflammatory chemicals also activate receptors on the neutrophils, which attach tightly to the selectins on the endothelial cells. This causes the neutrophils to stick to the endothelium and stop rolling. This accumulation of neutrophils along the walls of the capillary is referred to as margination. The inflammatory mediators released by the injured tissue also bring about changes in the environment that cause mast cells to degranulate and release histamine. Histamine causes vasodilation and an opening of the junctions between the endothelial cells, allowing fluid and leukocytes to leave the capillary and enter the infected tissue. The neutrophils now undergo dramatic changes in shape and squeeze through the endothelial wall into the interstitial tissue fluid. This process is called diapedesis. The neutrophils, followed by other types of phagocytes, are attracted to the damaged tissue site by chemotactic substances released by bacteria and tissue breakdown products. They ingest and destroy invading bacteria at the site.
Your macrophages will get to the site too. By the way, I know I make this mistake and every time I tell myself not to do it. It's endothelial layer of the capillaries. I keep referring to it as epithelial. So in your anatomy physiology, endothelial is the correct one. And I don't know why I know it and I just keep forgetting and keep going epithelial. Questions about inflammation. So this is what inflammation is, right? That's how inflammation starts. Anytime you have a signal that says there's damage, inflammation is going to happen. So histamine is the one that we're going to look at. So they are released by inflammatory inducing chemicals. How do you keep inflammation going? So histamines are going to release, mast cells will release it. Basophils that get to the site will also release it. So they're going to release some more. Damaged tissue, as long as you have the cytoplasmic materials and they haven't been cleaned, they're going to signal inflammation. Eosinophils, neutrophils, macrophages, they will constantly release this. If they keep getting the same signal that not everything is well, they will keep releasing these and making sure inflammation goes on. Okay? The second things go back to normal, then inflammation can stop. But your inflammation will continuously happen. And the result is non-specific. Meaning, as you saw, it doesn't matter if the bacteria is there. It doesn't matter if you just cut your skin. You cut your epithelial cells inside your mouth. You took a little tiny bite. You scratched yourself inside. A bacteria that you took with your food is going and it's damaging your colon. Until they get the signal, up to the point that they keep getting the signal, inflammation is going to continue and it's non-specific. These are the cells that are involved in the inflammation. Basophils, remember these are chemicals that they just release chemicals that keep going inflammation. Eosinophils, neutrophils, mast cells. Macrophages will get there too. These are your phagocytic cells, they're the top ones. At first you will have a lot of neutrophils. The good thing about neutrophils is they're tiny and they can get into the tissues. Sometimes your inflammation actually gets activated because a macrophage is activated. So let's say in the, your colon, you have a lot of macrophages in that area. Just because a bacteria is there and your macrophages are going through and they're engulfing a lot of them, going through phagocytosis, that macrophage is going to set off an alarm too. So it doesn't always have to be a damage to your mucous membrane. Any of these cells, they think, I have something to fight, they will set off the alarm and inflammation happens. Remember natural killer cells, I said they're T lymphocytes. I'm not going to ask you guys about this, but they are T cells. And we always say T cells are in your specific immune response. Natural killer cells are your non-specific. They fight against virally infected cells and cancerous cells. It is thanks to natural killer cells that majority of people are not constantly getting cancers. Mutations happen in our systems and you can create cancerous cells. But in most cases, natural killer cells find them first and destroy them before they can cause a problem. How they got activated and they're not specific, it's kind of like, and like I said, I won't ask you about it, but it's interesting to know because a lot of people are now interested about cancer. So the natural killer cell is going around and on their surface, they have a series of buttons, series of receptors. Some of these receptors are positive, good receptors, and some of them are negative receptors. Positive receptors mean everything is good. If you punch these in the proper order, that means you know the security code for me and you're setting the alarm off. The alarm is off. So your normal cells, constantly when natural killer cells go to them, the positive buttons are being pushed. So then they step back and they're like, oh, okay, perfect. You know the passcode. So you're good. You still know the codes, the secret code, you're okay. But if they go and instead of, sorry, I always reverse that, negative and positive. So the negative meaning as far as like, negative is shut in, so negative reaction. Natural killer cell shuts down and says, you're good, we're going. If the wrong buttons are pushed, so let's even forget about negative and positive, is the wrong receptors are pushed and none of the right receptors are triggered, that are healthy receptors are triggered, then that natural killer cell says, you put in the wrong code. So that means you're not normal, I gotta kill you, and they start killing it. So that's what they are. And they will do that for cancerous cells, any type of virally infected cells. They don't care that it's this kind versus that kind. Outcome of inflammation. Why would you have inflammation? First and foremost, you've damaged tissue, you are repairing tissue. Inflammation sends all these extra stuff there so your cells can divide and repair the tissue. Second, most important thing, especially for infectious diseases, is phagocytosis. You want to remove the infectious agents, you want to remove damaged cells and allow for repairing and going back to normal. So let's look at the process of phagocytosis. This is actually an image of your phagocytic cells, macrophages, that are actually taking in this tumorous cell. They're engulfing it. They're going to eat it up. As like anything else with any other white blood cells, cells that are involved in phagocytosis, your two cells are neutrophils and macrophages. They have to go to the site where trouble is happening. They do that by chemotaxis. So how they get to the site is by chemotaxis. They respond to inflammatory inducing chemicals and they go to the site. Next thing, they have to find the pathogen and adhere to it. And in these cases, they use PAMPs, pathogen associated molecular patterns. It's non-specific. Anything that is not supposed to be there, they go through phagocytosis of that. Even damaged cells, they will go through phagocytosis of those debris. You're not supposed to be found outside of my cells, I'm going to remove you. Your damaged cell membrane, I'm going to remove you. So phagocytosis. What I'm going to use is a macrophage. Why? Because macrophages ultimately speak to your third line of defense. So chemotaxis happens, they get there. So this is my macrophage. And let's use the virus, since we used the virus last time. So we used the same virus. They come there and the virus has all these antigens on its surface. Okay. The reason I'm drawing this is because the animation doesn't have some of these extra information about macrophages that I want you to have. So your macrophage is a nucleated cell. It has the nucleus somewhere in there. As it is your nucleated cell, it will have MHC1s, and those MHC1s will have your autoantigen. So I'm just going to say autoantigen and draw it like that. So you're going to have a ton of these MHC1s. This macrophage also in it has, and let's draw that as regulars. Little organelles called lysosomes. Lysosomes are small vesicles that have severe, like very strong digestive enzymes, right? Macrophage gets to the site, and what it does, it finds a virus in this case, and it's going to go through the process of phagocytosis. Phagocytosis is a form of endocytosis, where the cell is going to use the membrane to engulf something. So when it gets there, what it's going to do is it's going to try to either push this against something and using cytoplasmic membrane and cytoskeletons and all of that, it's going to extend its membrane around it, pseudoarms, and wrap it inside. So it will be something like, you know, it will just go covered from all areas. So the membrane surrounds it. Once it surrounds it, it's going to come in a little vesicle, so pinches it off, engulfs it, comes in a little vesicle. We refer to this as phagosome. So phagocytosis, once the pathogen is inside, we refer to this vesicle as phagosome. Having phagosome inside triggers the lysosomes to go in. So next, you're going to have phagosome and lysosome fused together, and they're going to release all of their material in there. So they're going to start breaking down the virus. This is referred to as phagolysosome. It's going to go and break it apart. In macrophages, in neutrophils, this is it, it's done. In macrophages, what they do is then they actually will send a little tiny vesicle to, this is Golgi complex, a little tiny vesicle with the antigens they're going to send that here. That antigen is going to be, your Golgi complex will recognize that this is a foreign antigen. It's going to attach it to MHC2. And send it to the surface. So then they're sent to the surface. This is MHC2 with pathogenic antigen or exogenous. MHC1 will always present antigens that are made in the cell. MHC2 will always present antigens that were coming inside, were taken into the cell from outside.
So for the phagolysosome part, they eat up or kill everything besides its antigen.
Yes, they will. They basically break things apart,
Okay.
and then they will choose. I'm going to break this a little bit more. In the case, they will actually send some of these antigens to the Golgi complex to present.
Thank you.
At this point, your macrophage will turn into what we call an antigen presenting cell. or APC. Jason.
So only macrophages present on MHC,
Yes.
so like a neutrophil wouldn't?
Neutrophil does not have MHC2. So the only ones that have it are macrophages and B cells. But we'll talk about B cells later on and why they do that. So this is what a macrophage does. A neutrophil just goes and breaks everything apart. And the phagolysosome that is made in the neutrophil totally breaks everything apart, gets rid of the waste, anything the cell can use, keeps it, does not become an antigen presenting cell. This macrophage is an antigen presenting cell. This guy is going to communicate to your third line of defense if it needs to. If everything is removed and things go back to normal, your inflammation dies, and these guys are going to basically calm down and eventually shut themselves down, remove the MHC2, everything, and just go back to like a macrophage that is waiting for the next thing. But in many cases, as far as the infectious diseases, that doesn't happen. So they have to go one step further, and they have to talk to your third line of defense. Something else that I want to mention here that the animation actually talks about is a term called an opsonin. Opsonin or opsonization. Sometimes the process of phagocytosis is hard. The pathogen is slippery. It tries to push it in, it slips by. In some cases, your system actually has certain molecules that, in addition, during inflammation, for example, it will produce these and they can attach to the surface of pathogens. And the macrophages will have receptors on them, so they will have a receptor on the surface, which is an opsonin receptor. Opsonin is kind of like put a handle on a bacteria. So a bacteria that is slippery, opsonin is a protein that will attach to the surface of that bacteria, and the receptors have the hook for it on the macrophage. So this macrophage doesn't have to push against something. The opsonin attaches to it. It's kind of like grabbing it and says, oh, I got you, pulls it in. Like sending a hook, grabbing it and bringing it in. Some opsonins are produced, these are proteins, inflammatory proteins that are released. Like the example that you have in your animation calls C3b. It's a protein, complementary protein. I will tell you guys about it. You're going to hate me for it, but we're going to talk about complement proteins. Antibodies are opsonin too. But antibodies actually happen third line of defense. And this is why I say immune system is funny. Antibodies are formed there. So a macrophage actually signaled your third line of defense. But the third line of defense, the antibody actually now comes back and helps macrophage to remove more things because it's an opsonin. All right? So let's take a look at that. By the way, look at this. This is a macrophage. This is a bacteria. Keep an eye, remember these little things here? This is associated with complement system. It looks like there's a hole there, right? And these other forms, little borders and holes are starting to show up. This is function of complement system, which I'll bring upon you right after this. By the way, this lady who goes through this narration, she goes phagocytosis and phagocytosis. She calls it phagocytosis, phagosome, and then it goes through phagosis or something like that. So she just totally switches back and forth.
Several white blood cells, such as neutrophils and macrophages, are involved in an innate immune defense known as phagocytosis. In phagocytosis, these phagocyte cells are attracted to the area of microbial invasion by chemical products of the invading microorganism, chemical signals released by injured host cells, or by components of the complement system. The phagocyte moves into the area of invasion and then attaches to the microorganism. The attachment is mediated by a variety of surface receptors. For example, the C3b component of complement coats bacteria or other particles and then binds to C3b receptors on the phagocyte. This process of coating to enhance phagocytosis is called opsonization. The microorganism is then engulfed by the phagocyte into a vacuole known as a phagosome. Vesicles in the cytoplasm called lysosomes fuse with the phagosome, releasing digestive enzymes such as lysozyme and proteases into the phagosome. The structure resulting from this fusion is called a phagolysosome. Inside the phagolysosome, microorganisms are killed, then digested. Finally, the digested contents of the phagolysosome are eliminated from the phagocyte by exocytosis.
So this one pretty much covered everything except it doesn't show you MHC1s and MHC2s for macrophages. Neutrophils, basically this is what they do, and that's it, they leave it at that. Questions regarding phagocytosis? So far, again, these are some of the images, like dendritic cells actually can send one of their dendrites, kind of wrap it around the bacteria like a lasso and then pull it in. Now, up to this point, inflammation, what we've talked about, is involved in cellular responses. So white blood cells getting there, white blood cells doing this, white blood cells doing that, macrophages, neutrophils. The process of inflammation will release some non-cellular or chemical responses also. The three we're going to look at here are complement proteins, how they react and how they fight, interferons, and defensins. First, let's look at the complement proteins. Complement proteins are referred to as complement cascade responses. This is because you have a large group of complement proteins, and I'll write them down. Together they function, and they work in a cascade way. Like one triggers the activation of the other one, that one triggers the activation of the third, the second, the fourth, and all of that. Eventually, the complement response is triggered. The purpose of complement response is this. It will create what we call a MAC, membrane attack complex, that basically, like you saw in that image, creates a hole in the membrane, in the surface of the pathogens, which again, kind of, when you have a hole in a cellular pathogen, that pathogen is going to go through lysis. Complement only works on cellular pathogens. So complement cascade will not work on a virus. Will only work on cellular pathogens where it can poke a hole and lyse them, break them open. And the hole will go through the whole entire cell wall, cell membrane, all of that stuff will go through it. When they looked at complement pathway, they used to think the only way it got activated was by this method called classical pathway. So they thought, okay, complement gets activated during the third line of defense when you have your adaptive immune response and when you've produced antibodies. Then when they looked at immunocompromised individuals who are immune system, the third line of defense, like people who had AIDS, complement was still triggered. So they realized complement can actually get activated during inflammation too. So now they have two pathways. classical or alternate or alternative pathway. We're gonna look at these two pathways. Right now, I'm just gonna look at alternate pathway because I have not told you about how antibodies are produced or anything. So, we're gonna look at alternate pathway. So complements is a group of proteins. These are the proteins that we have in the complement system. A group of proteins called complement proteins, short we call them C. Complements proteins 1 through complement protein 9. So C1, C2, C3, C4, C5, C6, C7, C8, C9. Okay. We have a protein called factor B. We have another protein called factor D. And finally, a protein called Pro-per D. These are all the proteins that you could have as complement proteins. And they are found naturally in your plasma. So they're proteins that are naturally in your plasma. They need to get activated, and in the pathway is basically one gets activated, activates the other, and so on and so forth. That's why we call them a cascade. In the alternate pathway that we're going to look at, so we're going to first look at the alternate pathway. This happens during inflammation. So it's with the help of inflammation this is going to get triggered. Proteins that are involved in this are proteins C3 to C3, C5 through C9. So C3, C5, 6, 7, 8, and 9. Factor B, factor D, and Pro-per D. I'm just going to write it short. One other interesting fact is complement protein 3, complement protein 5, factor B, when they get activated, their activation is the split of the protein. Factor 3 has two portions, 3A and 3B. Same thing with 5, two portions, 5A, 5B. Factor B has BA, BB. This is why I'm like, you're gonna hate me. Key thing, part B's are important. Part A's have like side effects, but in the cascade turn on, part B's are the one that are used. So to be or not to be, B is the answer. All right. Okay. When you're looking at this, forget about B and A and all of that. See the order of the proteins, then implement part B and A and all of that, because the names are crazy. Okay. So, alternate pathway initiates with complement protein C. This is what starts this process. If complement protein 3 is splitting and we get a lot of it and attaches to a pathogen, then alternate pathway gets started. But if it doesn't, it won't get activated. This is why they didn't know about the alternate. When it's at the beginning of inflammation, you don't have a lot of pathogens. And this protein 3 might not find a pathogen. So it's wishy-washy, whether it get activated or not. Obviously, after you have a lot of the pathogen, it will get activated. But by then, you might have the third line of defense. When you have, either it's due to inflammation or naturally some of it is like this, they don't know. Let's say about all the complement protein 3s that you have, about, I'm not sure if it's 1%, but 1% of them naturally split into C3A and C3B. When inflammation happens, remember plasma is going to the site of inflammation. So all of these complement proteins are going to that site of inflammation. Why? Because plasma is leaking out. So their concentration is going to be high. This complement is against cellular pathogens. So this is my bacteria. Okay. I'm using bacteria because it's easier. So I have a bacteria, pathogen is there. C3A and C3B. Let's make them look like. So this is A part, this is B part. Okay, so this is A and B. They split, so now in the cell I have 3A and 3B. If one of the 3Bs by chance collides with the bacteria, it will attach to the surface of that bacteria. So 3B will attach to the surface of the bacteria. If it doesn't, it will shut down. Or if it attaches to your cells, it shuts down. It has to attach to the bacteria. So now you have C3B attached there. That C3B, when it's attached, it's gonna call in a factor B. Factor B, also, let's do it this way. This is A, and this is B. So BA, BB. A whole factor B is going to attach to this guy. But factor D is called too. Factor D comes and says you only need part B and removes the A. So you end up with a protein mixture on the surface that we call it a C3 convertase, and it's made up of C3BBB. C3 convertase is an enzyme that now that you have it, will split all the C3s in the area. So C3 convertase's job is to break C3 into 3A and C3B. Before you had 1%. Out of that 1%, maybe you got lucky and one C3B attached and it stayed active. If none of them find a bacteria, none of them would remain active. But once it's there, then it will get factor B. Eventually, with the help of factor D, it's just going to keep the part B of that factor B. So it turns into C3BBB. That is the name, or that's the formula for an enzyme we call C3 convertase. C3 convertase converts all C3s that are next to it into active, so splits them. Now you have 100% concentration of C3b. All of those guys are going to try to find a bacteria and attach to. Meanwhile, after a while that a lot of the concentration of C3b goes up, the enzyme properdine now comes and kind of touches them, changes their structure a little bit, and that allows for another C3b to get attached there. With that attachment, this becomes what we call C5 convertase. So the formula for C5 convertase is C3b, BB, 3b. The job of this one is to split C5 into 5A and 5B. 5B's come and attach to the surface here. When 5B is attached to the surface of the bacteria, it attracts one C6. C6 will attract a C7. C7 will attract a C8. That C8 will attract a number of C9s. They all insert into the membrane of the bacteria, attach together, and they create what we call a MAC, membrane attack complex. There's a MAC attack. This pokes a hole, this bacteria lyses out.
What does the C5 convertase do again?
Breaks C5 into C5A and C5B. Now, the parts that we have out, the C3A floats away, C5A floats away, but they have a function. If you have a lot of C3A, that's going to push for neutrophils to come to here. So C3A kind of is like an alarm for neutrophils, scream for neutrophils to come here. C5A pushes the inflammation to keep going on. Let me show you the animation for it. To make it simple, say C3 is important for activation of complement protein. C3 on its own naturally splits, or 1% of it gets activated. And how do they get activated? They split to A and B. If C3b finds a bacteria, will attach to the surface of it, or a fungus, it will attach to the surface of it. That keeps that C3b activated. It's going to call factor B. Factor B and D together will come. Eventually, the result of that is B portion of B protein will stay there. Together, these two together make C3b-BB. That's a C3 convertase. So now you're splitting or activating all of the C3s. Your concentration of C3A and C3B goes up. Some of those C3Bs will attach to the surface of bacteria. Some of them will come with the help of properdine attached to C3 convertase. They change C3 convertase into C5 convertase. C3b, BB, 3b. Once C5 convertase is there, you split C5s. They attach to the surface of the cells, called 6, 7, 8, and multiple 9s. They make the whole. C3A, C5A, they induce inflammation and bring neutrophils. C3B is an opsonate. All right, so let me show you the activation, the animation. I gotta go forward in it because the first part of it is showing you. I know, you think it's horrible, but once you do it a few times on your own, you'll see it's not that bad.
That's not gonna do anything.
The classical is easier.
Maybe later. I know.
So
It's a lot. Is there something like this?
All right, so this is the alternate pathway. All right, because they talk about classical first, they will talk about this is C3 convertase analog to C4b, C2b from the classical pathway. So kind of ignore that right now. Once I talk about the classical pathway, then it becomes even more clear. I know, I'm sorry, I could not find one that is just the alternate.
pathway.
Not require the presence of antibodies. Activation begins with cleavage of C3 into C3A and C3B, either spontaneously or via blood protein. C3b binds to foreign substances. Factor B, a blood protein, attaches to C3b and is cleaved into two fragments, BA and BB, by factor D, forming C3BBB.
So this is now C3 convertase. She's going to say an analog to or similar to C4B2B. That's classical stuff.
Analogous to C4b2b. Both structures, C4b2b and C3bbb, are called C3 convertase.
All convertases' job, whether it's C3 convertase or CR5, is to split. The C3 convertase splits C3. C5 convertase splits C5. When they are split, that means they are activated.
C3 convertase rapidly cleaves more C3 into C3A and C3B. C3 convertase is further stabilized by property, which allows another addition of C3b, forming C5 convertase.
So C3b, BB, 3b. That's C5 convertase. And she's going to say this is similar to C4b, 2b, 3b. Oh, he didn't say. All right. Now, next one.
Is it too late to say slow down?
I'm going to let you guys go after this animation. This one shows you the functions of it. Like, what is the purpose of complement cascade?
Regardless of whether the complement system is activated by the classical, alternative, or lectin pathway, the activated complement proteins are involved in several important processes associated with host defenses, including inflammation, attraction of neutrophils, opsonization.
I think I said it the reverse. I said C3A brings neutrophils. No, it's reverse. Inflammation, 5A neutrophils.
And cell lysis. Complement C3A participates in inflammation by inducing changes that cause the walls of local blood vessels to become more permeable. This facilitates movement of phagocytic cells from the circulation into the tissues.
So they're going to keep inflammation
Complement
going.
5A induces a directed chemotactic migration of phagocytes to the site of complement activation. Complement C3b functions as an opsonin. It binds to microbial cells, making them more susceptible to phagocytosis.
So having just C3b allows the phagocytic cells to just, it's like the handle. They grab that and pull it in. Now they don't have to worry about pushing it. It makes phagocytosis better, easier for them to go through the process of phagocytosis.
Complement proteins C5b, C6, C7, C8, and C9 react together to form a hole in the cell membrane. When C5 convertase converts C5 into C5a and C5b, the C5b binds to the cell surface and combines with C6. C5b6 binds C7, enabling it to insert into the membrane. Binding of C8 is followed by binding of several C9s. The hole generated by formation of this membrane attack complex causes the cell to lyse.
So that's what we call MAC, membrane attack complex. So are your brains fried completely? Sure? You don't want me to continue? All right, guys. So inflammation is not that bad. Keep an idea, but so the inflammation is going to go on. The alternate pathway, I know, it sounded like too crazy, but just look at the order of what protein comes where, then worry about this is splitting this is this, or just even know what is C3 doing, splitting to 3A, 3B. What is part A doing? What is the job of part B? Factor B, I only need BB part of it. Factor D, its job is to split factor B. Property makes it easier for me to make C5 convertase. And then look at the individual jobs of those. All right, on Wednesday, after our lab, which is gonna be pretty long, so plan on being here till like 3 o'clock, okay? Where we'll finish interferons and properdine and fever, which is not that bad, and then I will kind of start with the specific immune response and then go, I don't know how far I will get with that, but we'll have other class times for immune system. Okay? All right, guys. On